Hello students, in this video we'll study on Callus culture. If you study the history of Callus culture, it was first successfully done by R.J. Gothard from France in 1934 and to 1937. He was the first person succeeded in promoting the development of callus tissue from excised cambial tissue of Salix caprica, that is an woody plant, and other woody species. He was able to promote the growth of callus tissue using a simple nutrient medium, that is in vitro, supplemented with the vitamins thiamine, pyridoxine, nicotinic acid, and IAA, that is indole acetic acid. And the next person we remember is Nobocot from France itself in 1939. He was established the callus culture capable of potentially unlimited growth on semi-solid agar medium. He started his work using a taproot explant of carrot, and he was also able to maintain the culture by simply transferring the portions of callus to fresh media at a regular interval of 4 to 6 weeks. The next person, as we all know that in the in vitro condition in the laboratory we use MS media, the Scuse and Miller. They first put forward the concept of hormonal control of organ formation from from the callus tissue. They also suggested that the equal concentration of auxin and kinetin induce the continuous growth of callus tissue. An inclusion of kinetin in the callus media has made to initiate the formation of shoots. So if you define the callus is defined as an unorganized proliferative mass of cells produced from isolated plant cell, that is from explant. It may be from cells, tissues, or organs when grown aseptically on artificial nutrient media in the glass bottles or glass vials under controlled experimental condition. It is called as callus. Callus says these are the pictures of callus where here the shoot is initiated and the well differentiated the leaves you can see in this picture. That is, callus is defined as undifferentiated mass of cells. So what is the nature of this callus? The callus, which grows actively, may be composed of large vacuolated parenchyma cells and small closely arranged dividing cells. The such variations among the cells of callus tissue is due to the origin and the age of the callus and composition of culture media. And the Grant and Fuller, they are the two scientists, noted that these are that is a unorganized friable callus contains a larger amount of cell wall of polysaccharides than the compact callus, but a lower amount of cellulose compared with the hemicellulose and pectin substance. In a callus culture, phenotypic variations may occur due to the epigenetic changes. Such changes are reversible and occur due to the selective gene expression, as in cytokinin habituation. The callus and prolonged culture shows the changes in the nuclear cytology of the cells and changes both in chromosome structure and chromosome number also been noticed in the callus culture. Chromosomal aberrations and nuclear fragmentation of endopolyploidy, etc. There is also observed in the. See here, this is again the picture of callus where it is the undifferentiated mass cells. They are initiating the formation of shoot. What is the principle of callus culture? For successful initiation of callus culture, three important criteria should be followed. The first one is aseptic preparation of plant material. As we all know that the plant tissue culture should be carried out under aseptic condition. Second point, the selection of suitable nutrient medium supplemented with the appropriate ratio of auxins and cytokinins for their root shoot initiation. And the third point is incubation of callus culture under controlled physical condition. And different plant parts carry number of surface-borne microorganisms like bacteria, fungus, etc. So before attempting to initiate the callus culture, it is necessary to surface sterilize the plant parts which are to be cultured. The typical plant parts may be the segments of root, a stem, the pieces of leaf lamina, flower petals, etc. The excised plant parts are called explants and are first washed with the liquid detergents and also fungicide, generally 5%. T-pol. T-pol is a fungicide. So, how this callus culture technique is followed? The first step should be surface sterilization. The explants are surface sterilized by most commonly used chemicals such as mercury chloride, sodium hypochlorite, and also sometimes we can use 70% alcohol also. Okay, after surface sterilization, the explants are repeatedly rinsed with autoclave distilled water to remove the traces. The surface sterilized explant is cut into small pieces under aseptic condition, okay, a few millimeter in size. And the size of the explant is critical factor for the induction of callus tissue. The explants are finally transferred aseptically on a suitable nutrient medium and solidified agar, which gives a like a substrate, which act as a the solid substrate for the media on which the callus is initiated. Agar solidified as semi-solid nutrient medium after its preparation, sterilized by autoclave at 50 lb pressure and 15 minutes, it is used for induction of callus tissue. The incubated culture under controlled physical conditions such as temperature, light, and humidity is indispensable for proper initiation of callus tissue. The suitable temperature for in vitro callus initiation and growth is usually 25 plus or minus 2 degree, and in some plant material, initiation and growth of callus tissue takes place in a totally dark condition. However, in case of other plant material, particularly photo period may vary, that is from 16 hours light to 8 hours dark, it is necessary for the initiation of growth of callus tissue. Approximately 2000 to 3000 lux artificial light intensity is needed, and cool light fluorescent lamps are generally used for providing light. Generally, 55% to 60% relative humidity is maintained in the culture room for initiation of callus. So these are the different photographs which is taken from the research papers where you can see the undifferentiated mass of cells and initiation of roots and well differentiated the shoot system and also the root system. The development of callus culture. Callus formation from the explants occurs in three stages. The first step it is induction stage. Metabolism is stimulated and the cells prepare to divide. The cell size remains unchanged. The cell division stage, second one is the cell division stage. The cells divide actively and the cell size decreases. Cell division is mainly periclinical and occurs towards the periphery, giving rise to wound cambial cells. And differentiation. The cells differentiate by expansion and maturation. That means when you inoculate the explant, it has to revert back to its meristematic stage. That process we call it as differentiation. Rapidly growing callus cells are more or less like a growth rate alike, and as the growth rate decreases, the callus show their characteristic structure and forms. But all the calluses have some similarities. They all contain a nodular and sheath meristems in the groups and scattered throughout the tissue. The vascular nodules and the meristemites are formed in the callus from a small group of meristematic cells, and the nodules may not differentiate, may form a root or shoot primordia or embryos. So this is how the callus culture technique is followed. That explant is taken from the pot or from the field. The next step is surface sterilization, and it is inoculated into the media. After inoculation, it is the appropriate condition are provided to induce the callus. So this is the undifferentiated mass of cells. Again, this callus may be induced to form the shoot regeneration. From shoot regeneration, you can regenerate the plantlets and they are hardened after, that is, shoot formation before leaving it in the field. In a flow chart, the explant source, the first stage is in vitro callus induction, second stage shoot regeneration from the derived tissue, the callus derived regenerated plantlets excluding root, and the third stage is rooting of regenerated plantlets, and the last one is completely developed plantlets which are rooted. Again, these are the pictures of callus tissue. So why we need to do this callus culture? What are the application of callus culture? So callus can develop into many structure by manipulation of hormones because you can handle it in the in vitro and aseptic condition, and you can manipulate by recombinant DNA technology also. You can transfer them into form a liquid media called suspension cell culture for secondary metabolite production, and it can be transferred to the cytokinase rich media, induction of shoots, advantageous shoot formation for in vitro regeneration. The callus culture can be used. And it can be transferred to auxin rich media where advantageous root system can be formed, and these callus can be induced to form somatic embryos. In turn, they are used for the production of artificial seeds, and transferred to media with no plant growth hormones to form the their variations, that we can observe the homoclonal variations also by this callus culture, where this can be disintegrated to form single cells, and then they are inoculated where we can observe some variations also. Okay, this is about the callus culture. We'll continue the next class in the next video. Thank you.